Welcome to quarantine learning of G protein coupled receptors. So here is our diagram. We have our little phospholipid bilayer made out of Q-tips, and then we have each of our special sections made out of paper cutouts. So to start this off, this is going to be for a G protein coupled receptor with an alpha S subunit. So starting off, you have your first messenger here. So in this case, we have the hormone that will couple with the receptor. And then from there, you have your subunits. So we have an alpha, a beta, and a lambda subunit. And for this one, we're focusing on the alpha S. So to activate the subunit, GTP will come in and take the place of GDP. So it takes it from an inactive alpha S subunit to an active alpha S subunit. You can see here with the GTP. And then this GTP will activate the effector. In the case of an alpha S subunit, your effector is going to be adenyl cyclase. And the adenyl cyclase will use ATP to catalyze the production of cyclic AMP or CAMP. And then the cyclic AMP will work on an inactive protein kinase. For the alpha S, it's protein kinase A, and it will help to transform it from the inactive form of the protein kinase to the active form of the protein kinase. And then from there, it'll culminate into whatever cellular response that certain protein kinase will code for. Okay, so now that we've gone over the alpha S subunit, we're going to go over what happens with the alpha Q subunit. So again, starting off the same way, we have the extracellular space. The hormone or the first messenger will come in and couple with the receptor. The receptor has the three subunits of the G protein, and those are the alpha, beta, and lambda. So again, your G protein will have the GTP come up and replace the GDP, leading to the activated G protein. And then your G protein is going to act on the effector, which for this instance is going to be phospholipase C. And what the phospholipase C does is it cleaves the membrane phospholipid phosphatidylinositol biphosphate, also known as PIP2. Not shown here, but we did show what it breaks into, and that is IP3, which is also known as inositol triphosphate, and diacylglycerol, which is the DAG. So what happens from here, you have your IP3 that goes from its cleavage up by the effector towards the endoplasmic reticulum, not shown from a Q-tip shortage, but that will bind to the endoplasmic reticulum and liberate the release of calcium into the cytosol, which is all in here. And then the DAG will remain associated with the cell membrane, so it stays up here. And that one will activate protein kinase C. So you can see right here, this is our inactive protein kinase. So for the alpha Q subunit, it's going to be protein kinase C. And then with ATP, that will become activated, and then it will go on to perform the rest of the cell functions, leading to the cellular response. So that is the alpha Q subunit of the G protein. Okay, so now here's a little bonus on a G protein coupled receptor that I found really interesting going through all of my classes with embryology that I now fully understand, which is really cool. So the G protein coupled receptor is Gpcr3, and it's found in the mammalian brain and oocyte, and it's known as an orphan receptor, which I find really cool. It doesn't have a specified ligand that they know that activates it. So it's potentially something in like the follicular fluid, but they haven't quite identified what activates this G protein coupled receptor. So I found that really interesting. So it's not very diagrammed as it would be in a cell, but just a kind of basic flow through. So we have the Gpcr3 right here with the three subunits, the alpha, the beta, and lambda, and then we have our little G DP right here, and then it's activated to GTP, where it acts on the adenyl cyclase with ATP, and then that promotes the upregulation of cyclic AMP production. And you see we have the inactive form of pKa right here. So that will, the high cyclic AMP with the inactive pKa will activate your pKa, and then the active form of pKa will act on MPF, which is the maturation promotion factor, hereby inactivating it. So the maturation promotion factor is actually inactivated by the active form of pKa. And then later, when the oocyte begins detaching from the cumulus cells, you'll have a downregulation of cyclic AMP, which will promote the inactive form of pKa, inactivating the pKa, and therefore activating your MPF, so your oocyte will begin to mature. So that was very interesting to go through this cycle and understand it fully.